match up with. So what do we have? We've got to go over mitosis and meiosis. So these can be a little confusing and they take a little practice. You have to go through and you have to be able to, one, know the differences between mitosis and meiosis. They sound the same, they look similar, but they're different. You also need to know the kind of the steps in the cell cycle. You also should know the steps in making a protein. So where do we start from? Let's talk about DNA. So DNA lives inside the nucleus, right? But what's DNA? It's all these base pairs, A's, T's, G's. We know the law of complementary base pairing. C's and G's go together. T's and A's go together. And that's all it is. Just this long 3.8 billion of those stretched out. But these things, this DNA, is kind of not it's not just thrown in there. You don't just take it and go like you had like fishing line and you just like tossed it in. What would happen? It would get all tangled up and we wouldn't really be able to find anything in there. So what we do with the fishing line is we put it around and we kind of spool it around things. And then what we do is we take those spools and we tighten those up. And then as they tighten up, they become other structures. And then as that tightens up, they become other structures. So it's important to know the definitions here of those things. I'll go over them in just a second. So you need to keep these definitions straight in your head. It's very important because we're going to say genes and chromosomes and people use them interchangeably, but that's not really how this works. So what is DNA? What is DNA? DNA, well, let's think about this for a second. We have proteins in our body. And what are the proteins made out of? The proteins are made of amino acids. And the amino acids are put together in a very specific sequence. So there's going to be like methionine, leucine, isoleucine, proline, valine, and they're all, you don't need to know that the order of them is not important, but what you need to know is that if this was a protein like hemoglobin, right? It works because of its specific structure. It's going to have an iron that's in here and it's going to allow it to bind to oxygen. Now, if hemoglobin doesn't have its structure, it will not work. And we see that in disorders like what? Sickle cell anemia. So if you've ever seen sickle cell anemia, normally a red blood cell looks something like this, right? That's usually how they draw it. It doesn't have a nucleus in it, but that's usually kind of how it looks. Now, what's the important thing for a red blood cell is it carries oxygen. It's pretty important and also carries carbon dioxide out. So sickle cell anemia, what happens to the cells is they look like sickles, like that. They kind of fold in on themselves. A sickle was a thing that was like on a staff like that, and it was a metal thing, and you could chop wheat down with it. That's why they call it a sickle cell. It looks like it. The problem with this is if you see this structure compared to this one, do you think that this is going to effectively carry oxygen? No. The other thing is it can get caught up into different parts of the body because it's like kind of hooked, so it can get caught in areas and create clots, and that's bad too. So why does this happen? Well, this happens because of one amino acid change. You change, instead of a proline, you get a valine put in. And then what happens is the structure falls apart. So the whole reason that I'm showing you this is to demonstrate how important structure is to the protein. And now what determines the structure of the protein? The sequence of the amino acids, right? The sequence of the amino acids is going to give hemoglobin a structure. But now the question becomes is where are proteins made in our cells? They're made in our cells. What organelle makes them? The organelle that makes them is the ribosome. So the ribosome is going to make your proteins. It is a factory. Like imagine a factory that's just making beads. It's taking an amino acid bead, putting another one together, putting another one together. It's like making a giant necklace. That's all it's doing. And it has to put the beads in the correct order. So how does it know how to do that? How does it know that I'm going to put methionine and then valine and then arginine? And how does it know it's going to make these peptide bonds? Well, it has to have instructions. And what are those instructions? Where do they come from? They come from your DNA. They come from your DNA. Those base pairs, those A's, T's, G's, C's, all that stuff, that's the code. You guys know what Morse code? Oh, D. There's no D. You know what Morse code is? Morse code's a set of dots and dashes like this. And you can have like dot. I don't know any Boy Scouts in here know Morse code. I don't know it. You can have a dot, a dash, and a dot. And each of these three will represent a letter. So you go dash, 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 and that would represent like an A. And then you can do, or like an H. And then dot, dot, dot would be like an E, and then a dash dot dot would be like an L, and then like a dash dot dash where we've got like a P, and it would spell things out. Well, if that's the way your DNA is, every three letters represents an amino acid. So your DNA is the instructions for making your proteins. You remember, we have probably 20, 30,000, I think 20,000 proteins or different genes in our body. We have like hundreds of thousands of proteins and enzymes. You saw that like all the chemical reactions in the body are done by enzymes, and those are made of protein. So we have to have all of this, not all of the DNA, but some of it, some of the portions of the DNA are going to code, they're the instructions for making all of your proteins. Now, DNA is a 3.8 billion base pair molecule. It's this long molecule. Not all of it is going to make DNA or make proteins. It's like I think I explained this before. It's like you look for a recipe online, and most of the recipe you get to is the person like describing how they went to the south of France with their significant other, and they had such a wonderful time. And every time a year we make this delicious dish for my. And you're just like, all right, person, just get to the recipe. The DNA is the whole website with all of that stuff on there. The recipe is just the stuff that you want. That's what's called a gene. A gene is a small sequence of DNA that is going to code for a protein. Okay, so now we need to make the protein. So how do we do that? Well, where is it? Where is it made? It's made in the cytoplasm. The ribosomes are out in the cytoplasm. So if we draw our little cell here, and the DNA is in here in the nucleus, and the endoplasmic reticulum is out here, and the ribosomes are out here. So we need to get the instructions out to the ribosomes. Now, do you take the DNA out of the ribosomes? No, you do not. Because why? You don't want to damage it, because that's how you get mutations. You get mutations, you can get cancer, or the cell can die. So this is like much like going to the library. You go to the library and there's one reference book. You can't take that reference book out of the library. You're like, I need the recipe for hemoglobin. And you have it right there. So you can't tear it out. That would be vandalism and stealing. You don't want to do that. So what do? You take it to the photocopier and you photocopy a couple of pages of that recipe. You don't need to photocopy the whole book. That'd be making all your DNA. You wouldn't need that. You just need to photocopy a little bit. And so that's what you do. So say just this little piece right here is the sequence that we need that's going to be the instructions for making this hemoglobin. Well, we just make a photocopy of it. And the photocopy is called mRNA. Now, photocopying one strand of that is called transcription. And what does transcribe means? It means to write something down or to copy it. So essentially all you're doing, you're still in nucleotide language. You're going from nucleotide to nucleotide. You're just transcribing it. You're copying one thing to the other. Then after you transcribe it, what happens is the mRNA, the instruction, the messenger RNA goes out to the ribosome and it says, hey, read this. This is the instruction manual for making hemoglobin. The ribosome reads it and it says, okay, first one is ATG. All right, ATG, that's methionine. And then tRNA brings methionine in. Then it reads the next three, CAT. Oh, okay, that's proline. I can't remember. It's been so long since I took biochemistry. I don't know the exact ones. I probably should get it straight for you guys. But you get the example. And that is called translation, is when you translate in nucleotide language into amino acid language. That takes place in the cytoplasm. Okay. So just, I was going to stop, but just to review. So when we are making proteins, we have to make a protein. Whatever protein's made out of, amino acids. But you don't just put the amino acids in. It's a recipe. You don't just like throw everything in one pot and let it go. No, you got to like saute the onions first, and then you sweat the garlic, and then you throw in the tomato sauce. Like there's a instruction manual. There's a recipe you got to follow, and to make the structure come out appropriate. So what is that recipe? Our DNA. We can't. But where is the proteins made? They're made in the ribosome, which is out in the cytoplasm. Where is the DNA? In the nucleus. You can't take the DNA out of the nucleus to the ribosome, so you have to make a photocopy of it. The photocopy is mRNA. So you copy these base sequences, this base triplet, and you make an mRNA strand. That mRNA strand has what are called codons. So this is called a base, these are called base triplets. The mRNA is going to have a codon. So you're going to have a U because there's no T in mRNA. You're going to have an A and you're going to have a C. And then mRNA, so that's transcription, is making the mRNA. It's going to leave the nucleus and it's going to go to the cytoplasm and then it's going to give the instruction for the ribosomes to make protein. That is called translation.